Stem cells divide to replace older and dead cells. This is a stem cell replicating its DNA to create kneed lung cells. Each strand is made up a sequence of 4 chemical bases, or nucleotides, represented by the letters A, C, G, and T. The 2 strands come together where complementary nucleotides meet. This means that where there's an A in one strand, there will be a T in the opposite strand. And wherever there's a C, there will be a G in the other strand. DNA is a molecule made up of 2 strands twisted around each other in a double helix shape. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. Before DNA replication occurs, a large protein complex binds with a part of the DNA molecule. Where this protein complex binds is known as the origin of replication. The next step in DNA replication is to separate the 2 strands. This is done by an enzyme called helicase and topoisomerase, along with single-strand binding protein to keep the DNA from reforming. The topoisomerase works to remove the spiral structure of the double helix while the helicase unzips the DNA into 2 strands. The separated strands each provide a template for creating a new strand of DNA. This marks the starting point for the creation of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. And to increase efficiency and speed, a sliding clamp protein binds with DNA polymerase. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously. The DNA polymerase adding bases one by one in the 5' to the 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old conserved strand of DNA and one new one.